morning, everyone. Good to see you here bright and early this morning. Hope everyone had a time to grab a bite because I think the buses are running a bit late today. We're going to try and keep to time, so without further ado, I would like to start today's morning's presentation. A gentle reminder, please, to just silence your phones so that we can enjoy today's proceedings without any interruptions. My name is Kaylee, I'm from the NRF, and we're very happy to have you here with us again today. So, welcome to day 3 of the summit. We're at the halfway point now. We will now start with the Young Scientist presentations. I would like to invite Dr. Yang Le, Deputy Department Head of the Institute of Materials Research and Engineering at A*STAR, to chair this session. And we have our 2 presenters for today, Dr. Cosnet Lerato Rametse and Dr. Xu Jianjing. So first, Dr. Yang, please. Our first presenter this morning, as I mentioned, is Dr. Cosnet Lerato Rametse. So she's a PhD fellow in immunology at the University of Cape Town. Please do give her a warm welcome. I'll hand the mic now to you.
Morning, everyone. Oh, there we go. Good morning, everyone. Thank you, Chair, for the kind introduction. And a bigger thank you to this organizing committee for affording me the opportunity to share my work with you today. My PhD characterizes certain aspects of the male genital tract at a basic immunological level with respect to HIV risk and susceptibility. And the aspect that I'll focus on today is the penile barrier function. I'll tell you now why it is important to learn about the basic science penile immunity. Although it's always important to acknowledge that a lot of progress has been made since the enrollment of antiretroviral treatment, as evidenced by the decrease in HIV-associated morbidity and mortality, HIV new infections remain significantly high globally, especially in South Africa. Therefore, it is important for us to focus as scientists to try and understand what mechanisms underlie HIV transmission and susceptibility mechanisms. It is well documented that the commonest mode of HIV transmission is by heterosexual intercourse, and that the highest HIV burden in any sex or age cohort is amongst women of reproductive age. As a consequence, a large majority of research to date has largely focused on HIV transmission mechanisms that are focused on male to female transmission and female acquisition in the female genital tract, with HIV transmission mechanisms through the penile exposure being one of the least studied aspects of HIV research. It has also been sufficiently demonstrated by randomized controlled trials that medical male circumcision reduces HIV infections by 50 to 60%, particularly in areas of high prevalence. This then indicates that the foreskin plays an important role, but also that the other parts in the penile anatomy also contribute, explaining the remaining 40% risk. Interestingly, knowing this significant risk reduction in HIV infection following medical male circumcision, to date, we still don't understand the mechanisms that underlie this observed significant reduction in risk. Furthermore, it is important for us to then explore these factors to see what could be underlying these mechanisms and potentially use these identified factors to come up with alternative preventative strategies within the males. In our project, we look to characterize the female immune factors that could potentially underlie susceptibility in the male genital tract, for one, and we explore, with respect to the penile barrier function, what could potentially change following circumcision. And we hope by doing this, we would then generate knowledge that could provide as stepping stones into identifying potential targets within the male genital tract for alternative non-invasive HIV prevention strategies. HIV transmission is known to be amplified by multiple factors, one of which is asymptomatic STIs. Asymptomatic STIs, according to the WHO, in 2022, over 1 million sexually transmitted infections were acquired globally. That is both in developing and developed countries. And majority of those are asymptomatic STIs. And as we know, asymptomatic STIs could be virally caused, and that could be things like herpes. And it has been shown that about 75 to 90% of people have asymptomatic STIs. Additionally, things like trichomonas vaginalis and also chlamydia can also be examples of STIs that are asymptomatically presenting in individuals. It has also been demonstrated by various studies that STIs increase one's ability to acquire HIV and to transmit HIV. Additionally, it has been shown that people living with HIV are more susceptible to acquiring STIs, classical STIs, therefore the bidirectional complex relationship. This is an important factor to consider while looking at trying to understand susceptibility within the male genital tract. Now, to give you a brief schematic anatomical demonstration of the male penis, shown here is an uncircumcised penis and going through erection, but this is to adequately show you how the anatomy works. Proximally is the shaft, extends into the outer and inner foreskin, the coronal sulcus with this junction between the glans and the shaft, and distally the glans and the urethral meatus. All these sites have unique immune environments that are all susceptible to acquiring HIV. Additionally, the male external genitalia arises entirely from the urogenital sinus embryologically. As a result, it consequently only exists or is lined with what we call the stratified squamous epithelia, which is shown here. The stratified squamous epithelia is part of the epidermal layer that is part of the skin that covers the penis. And it's been shown in literature that this epidermal layer, which consists of 5 main stratas, that only the top layer is important in defining what we call barrier function in the skin. Therefore, what it implies is that maintaining the integrity of this layer is very important in ensuring that it performs its function to prevent host invasion by microorganisms such as HIV. There are a few models that have been, models of measurements that have been approved or accepted to act as proxies to measure barrier function, one of which is what we call transepithelial water loss, which I will refer to as TWEL as the presentation goes on. And essentially, what these models do is that they can be measured using devices as a proxy to define barrier function. TWEL, which is transepithelial water loss, is one of the key indicators for a good skin integrity. And it's been used vastly in literature, particularly in dermatological studies, to define barrier function. This is a model that we will use in our study to measure barrier integrity in the foreskin and the penis. Yes. And finally, as I mentioned, that the stratum corneum is the main layer within the epidermis that defines barrier function. Two of the main components that ensures this function is performed are the organization of the cells themselves, which is the stratified squamous epithelia, but also what we call tight junction proteins. The skin consists of many proteins which are called tight junction proteins, but one that has been shown in literature to be very important in defining and making sure that the integrity of the skin is intact is what we call claudin-1. It has been shown by Furisi and others in literature that when they knocked out the claudin gene in mice models, they died immediately after birth due to severe dehydration and compromised integrity of their skin. Now, having said that, our study main objective with regards to this aspect of the immunity function in the male genital tract, we seek to look at the barrier function both in the in vivo and ex vivo level of the penile anatomical sites, and we also seek to see how this is affected by the presence of asymptomatic STIs and how circumcision status affects barrier function. Could circumcision alter barrier function in the penile genital tract? We'll find out as I talk you through this presentation. Just to give you a brief overview of our methodology, around 720 male participants were recruited, and initially with this we collected first-pass urine samples and foreskins. These were then used to screen for common STIs as noted on the screen. Surprisingly, the screening revealed almost 20% prevalence of the different STIs. These are at the different regions in the Western Cape, South Africa. This data was then used to select 2 clinics that had the highest STI rates in order to do our primary study measures. From this subset, 203 male participants were then enrolled and recruited into the study, and what we call meter devices, which are often used in dermatological studies, which are then uses modalities such as transepithelial water loss and surface hydration, were used to measure these modalities at different sites in the penile genital tract, that is the shaft, the foreskin, and the glans. As I mentioned, all these sites are potential sites of HIV acquisition. Additionally, we had collaborative studies that were done in Chicago, University of Northwestern, and they collected some data relating to barrier integrity within males who were circumcised at birth, as opposed to the ones in South Africa. It's adults who actually decided to get circumcised at the adult stage, so we were able to measure before and after circumcision, whereas this cohort was a cross-sectional analysis where they just measured post, however many of years post circumcision at birth. And again, alongside all these, we also collected foreskins from which we extracted RNA, which were then used to perform qPCR for common tight junction proteins. Just to give you a quick summary of how the devices work, I'm not going to go into the details because of time, but the first 2 devices essentially, as the vapometer and the moisture SC, they measure dynamics within the stratum corneum only, and the other 2 goes all the way deep into the dermal areas. Important to note is that as part of taking the meter measures, we also measured the environmental conditions, such as the relative humidity and temperature, and these showed a strong correlation between the readings and the meter readings, and therefore this was considered when we did the final analysis. In terms of results, this first slide shows you the baseline characteristics. Now, what we did is, as I said, when we measured the different anatomical sites on the penis and using this different meter machines, we looked at the different modalities. And important to note, with the vapometer, which I noted to be the gold standard, we found that in pre-circumcised penises, when we measure at the glans, inner foreskin, and shaft, there was a significant difference between the T-wall readings in the glans and the inner foreskin. The inner foreskin had the highest T-wall reading, which means that it had a less or a compromised or less barrier function compared to the shaft. We also noted that this was followed by the glans, and again, it showed that there was more hydration in the different parts of the penis compared to the shaft. We then again looked at this on a molecular level, and we did qPCR for these various tight junction proteins, and what we found largely shown in the red as the inner foreskin, in the blue is the outer foreskin, and we show using qPCR that majority of these genes are significantly expressed in a lower level in the inner foreskin compared to the outer foreskin, which could be a potential mechanism explaining why the inner foreskin has been described with in vitro infections to be more susceptible to HIV infections as opposed to the outer foreskin. But what was interesting in this case was that if you look at Claudin 1, which is the main tight junction that defines barrier function, there was no significant difference between the inner and the outer foreskin, as well as filaggrin. I'll get back to this when I go into another part of my results. So, firstly, here we looked at the impact of circumcision. So, looking at the cross-sectional analysis at the Chicago cohort, what we did was that we compared these modalities between the circumcised and the uncircumcised. And it was very clear that when you looked at the circumcised and uncircumcised, the uncircumcised showed that the glans, which was consistent with what we found in South Africa, had higher transepithelial water loss, meaning that it was less, the barrier function was less intact compared to the shaft. But in the circumcised, as you can see, there's a significant difference in a shift of this measurements in the glans. We noted that there was no longer a difference between the shaft and the glans, indicating that circumcision improves barrier function in the glans. Secondly, we also did a longitudinal measurement, although the follow-up was very limited due to COVID, but also socio-behavioral factors within males when it comes to health-seeking behaviors. But what we saw, what was interesting was that when you looked at the T-well, We followed up the measurements at three visits time points, that is 2 weeks, 3 months, and 6 months post-circumcision. At 6 months post-circumcision, there is a significant decline in the T-well reading, which then suggests that what we noticed to decrease in the glands actually happens way over time following circumcision. We also looked at the impact of asymptomatic STIs on these measures, and what we found was that there was no significant difference in the T-well readings, but we found that in the stratum corneum there was increased moisture, which could implicate many things. I can't make valid conclusions, but we can say for sure that it will affect the microbiome and the kind of, microorganisms that sit in the different anatomical sites, and that could contribute to harboring an environment which could be friendly to HIV infections. This is where I show the significant difference here. And therefore, it shows that the presence of an asymptomatic STI alters the barrier function of it does not really alter the transepithelial water loss, but it increases the moisture in the apical epithelial layer. Finally, we also looked at the impact of asymptomatic STIs on the expression of molecular molecules that affect or influence the expression of barrier function within the foreskin. And what we see that is interesting in this case is that Claudin-1 now shows a significant difference in expression between the inner and the outer foreskin when there is an asymptomatic STI, suggesting that the presence of an asymptomatic STI not only increases moisture within the apical layer, but it also compromises the barrier function, rendering the different sides, particularly the foreskin, more susceptible to HIV infection. So in summary, we've shown that prior to medical male circumcision, the penile glands in the inner foreskin have significantly lower barrier function as compared to the penile shaft. The glands in the inner foreskin have significantly increased water content and moisture, which could have implications in terms of the kind of environment it creates for microorganisms. We also show that medical circumcision at birth results in an improvement in barrier function within the glands, and has been shown in literature that the glands is one of the most HIV susceptible structure within the penile genital tract. So this is interesting that we show that medical circumcision, one of the underlying mechanisms would be the improvement of barrier function within the glands. We also show that Claudin-1, which is significantly important in defining barrier function, is affected or is the expression is decreased in the foreskin in the presence of an asymptomatic STI. And therefore, these findings could be linked to HIV susceptibility mechanisms in the uncircumcised males and the presence of STIs. These could start giving us hints into potential targets in developing alternative preventative strategies, such as creating microgels which could target certain elements of barrier function defining factors. These are my references, and I would like to thank firstly my supervisor, Professor Clive Gray, and all the participants that were recruited and volunteered to be part of the study, our collaborators, and my funders. I'm happy to take questions. Thank you.
Thank you, Dr. Rametse. Can I invite Dr. Young now to moderate the Q&A session?
Thank you so much, Dr. Rametse, for the very interesting talk on the barrier functions of male genitalia against HIV infections. It's a very interesting topic. I hope you're all still bright-eyed and bushy-tailed at 8-something a.m. in the morning. Shall we have some questions, please, from the audience? Please don't feel shy. And those of us who are online, joining us online at this hour, please feel free to send in your questions on the online platform, that's Slido. All right, any questions, please, from the audience? Oh, yeah. Oh, sorry, I didn't see that. Yeah, sorry. Please.
Thank you so much, that was great. I'm really impressed at the data that you've got, the fact you can follow individuals pre and post circumcision, so I was wondering how you went about the recruitment for that.
So we formed partnerships with, so in Africa, medical male circumcision is a free service, first of all. So there are circumcision clinics that are linked to either non-profit organizations or governmental clinics. So we formed partnerships with those clinics. And usually post-circumcision, all the males are supposed to come back 2 weeks post-circumcision to do their checks. So that's how we got them into, to get the follow-ups. But moving forward, they got compensation for following up, although we expected way more numbers in terms of follow-up, especially at the 6 months post-follow-up, which again shows, because it's well known that males often, when it comes to health-seeking behaviors, they're quite hesitant. But I honestly think that it's mainly because of compensation that some of them came back. Yeah.
Thank you.
Thank you. Anyone else from the audience, please? If not, I will attempt to ask a question. I hope I'm not going off tangent a bit too much. But I'm wondering what's the impact on, say, the exposure to the barrier functions when they are exposed to the female genitalia? Are they, for example, in terms of preventing infection or transmission, is there an implication there as well when exposed to the female counterpart, female genitalia?
I mean, I wouldn't speak on barrier function specifically, but we know that when the male penis is inserted into the female vaginal tract, there's exposure of genital fluids, which would then affect inflammation of both the tissues, both the penile, different anatomical size, but also there's the mechanical pressure on the penis itself, so that could also kind of disrupt the integrity of the skin there, but that's all hypothetical.
I see. Interesting. Do we have more questions from both our online audience and our physical audience here? Yes, please. Yeah.
Hello? No, I was just wondering what are the future plans or directions of the...
I mean, I still have, today I just presented an aspect of the data, but we've done a lot of, we've done RNA extraction, so we've done sequencing, so we're still going to look at the changes in pro-inflammatory cytokines. We've done digital spatial profiling, where we looked at how the presence of STIs affects the different expression of pro-inflammatory cytokines on specific regions in the foreskin. And I think once, we have all that put together, that will kind of tell us where to go moving forward. But the plan is to then look at the key markers that are affected by asymptomatic STIs and try and do ex vivo infection models to see at an infection model level how is infection really affected following,
Thank you very much. Well done.
Thank you. But following on to the future directions, you mentioned at the end, for example, developing gels for further treatment preventions. Is there any specific properties of these gels? I'm coming from a materials point of view. What kind of properties are we looking for in these gels?
To be honest, I haven't thought about it to that extent in terms of the properties of the gel. I just know, I think the interest would be, what are you targeting? And then I guess would have to have more research and collaboration with, I guess, material scientists as well, to then figure out, based on the properties of the actual genital tract and the pH levels and all those other contributing factors to see. But essentially the interest is to firstly identify what exactly you'd like to target with the gels, of which in this case, I guess we can suggest that looking at tight junction proteins would be something that's useful, seeing that we've seen that there's a significant change following circumcision, which could be one of many mechanisms that could be explaining this observed reduction in HIV infection risk following circumcision.
All right, thank you. That's very good. And we've got one online questions. I hope it's showing up on the screen here. What is the magnitude of contribution of barrier function in increasing the susceptibility to HIV compared to other risk factors such as M2M, sexual hygiene, and so on?
I don't think I can confidently honestly say one has more magnitude compared to the other. The only level I can comment to at this point is that we found that barrier function is one of the mechanisms that contributes to susceptibility. And of course, things like sexual hygiene, we did take history when we did our barrier function our barrier function readings to put that into account. And we know that with man-to-man sexual transmission, the commonest way of acquiring the infection is by rectal exposure. So the epithelial layer lining the rectum is way more susceptible to HIV infections compared to the genital tract. So that's a different conversation on its own because the structure is different, but the susceptibility level is way more higher in man-to-man exposure because of the lining of the rectum.
I see, I learned a lot of new things today. Due to time constraints, let's see if we have one last question. Oh, yes, please. Yeah.
Great presentation. My name is Oliver from Australia. My question is about compliance and accessibility of potential therapies. So your research has potential implications on development of new therapies and treatments for people with HIV. So what are your thoughts on how that would actually be implemented so that people have access to those therapies?
I think that especially now, there's a lot more hesitancy towards circumcision compared to when it was introduced, especially in developed countries, but even more now increasingly in developing countries. So I think that in itself, coming up with non-invasive alternative preventative strategies would be probably encouraging to a lot of people. And I mean, in terms of access, to be honest, I mean, I think you were here yesterday when we spoke about many factors contributing to access, and that would have to be on a policy level, what local government would be willing to provide freely for the locals, and socioeconomic factors. So I think it's very complex, but yeah.
Okay. All right. Thank you. Maybe. Oh yes, please. Yeah. Maybe one final question.
Wonderful talk. I was wondering, in terms of circumcision, is there like a one circumcision for all the participants, or are there like several types of different circumcision, and if that might influence the results?
Well, I wouldn't say type. Well, in terms of types, it would be medical and traditional, because we have done a study in, Papua New Guinea populations actually, where we looked at the impact of traditional circumcision on barrier function. I just didn't discuss the data here, but it also kind of has the same impact on barrier function. But in terms of medical circumcision, the only differentiation is the technique that's involved. There's a dorsal slit circumcision, but essentially it does the same thing. It removes the foreskin tissue, and the healing would be the same. So I don't think that would affect, the effects, unless if you don't remove completely the entire foreskin, then of course you still have a piece of tissue that's still highly susceptible to being infected.
Thank you. I know you've been standing there. Maybe just a quick one. No, just a quic one. Yeah.
Okay, thank you for the presentation. My question is that normally HIV and other STI has like a bidirectional impact, synergetic impact. So can we generalize for other STI infection, because we focus is on HIV? Can we generalize the better impact for other STI infection? This is my one question. The other question is that there may be the level of hydration may be vary based on age. Maybe the level of hydration, because the water content of human beings differ based on the age. So how just have you assessed based on the age, maybe adolescent, young, is like? Thank you.
I didn't hear the first one. Did you?
I didn't as well. Could you repeat?
Because you focused on HIV, so can we generalize for other STI infections? Yeah.
Oh, you mean in terms of the improvement in barrier function affecting the other infections?
Yeah.
I don't think I can confidently say that, because they have different mechanisms to infecting the different anatomical sites of the penis. What I can say is that they influence each other in terms of increasing each other's susceptibility, but in terms of what decreases susceptibility in HIV, I can't confidently say it decreases with other STIs. Yeah. And with the water content and age, it's a good question, because when we did our data analysis, we did try to see if maybe there was a correlation between the age and the barrier function readings, but with our cohort, there wasn't really any significant correlation, but that definitely maybe could contribute to the time it takes for one to have the changes that we see based on age.
Thank you.
Thank you. Due to time constraint, I'll have to wrap this up. Thank you so much, Dr. Rametse, for the very interesting talk and discussion with us. Thank you. Yeah.